Hi everyone, my name is Eric Torres. I am a marketing manager at Biodium, and today's on-demand webinar is entitled Chemical Advancements of Modern Fluorophores and How CF Dyes Light the Way for Life Science Research. And so the focus of today's webinar is really on your common fluorescent dyes that are used in everyday research, this being your FITC, your rhodamines, your Cy or cyanine-based dyes, and a really particular focus on the chemical properties of these dyes and how they really advanced and evolved over the years to optimize performance, and how Biodium CF dyes really represent a major breakthrough in modern fluorescence. And so before moving on, I want to explain a little bit more about our company. So I work at Biodium, which is a life science reagent company, and we specialize in making innovative fluorescent reagents for life science research. And we're a small company that's only about 30 to 35 people. We were founded in 2001, so about almost 20 years, and we're based in the San Francisco Bay Area. We have worldwide distribution channels, and since our founding, we've patented numerous novel technologies. Today, I'll speak much about our CF dyes, but you may have also heard of our gel red and gel green nucleic acid gel stains, our apoptosis substrates NucView, our PCR evergreen dye, true black background reducers, and many more. And so our mission at Biodium is really to provide innovative solutions for scientists in life science research, specifically fluorescent life science reagents. And we do this by our strong collaboration and communication between our chemistry and biology groups, where our biology team, which encompass PhDs and our customer support, R&D, as well as marketing, to understand the problems that our customers face today and really communicating that to our chemistry R&D team, who can from the ground up build a molecule that will ultimately be applied to solving specific research problems today. And so let's start at the very beginning of the history of fluorescent dyes. So over 150 years ago, a German chemist, Adolf von Baer, was actually the first one who discovered the first fluorescent pigment. And he did this by synthesizing what he aptly named resorcinylene, which came from phthalic anhydride and resorcinol. But we know today this dye more commonly known as fluorescein or FITC, which is a xanthene-based fluorophore with a really strong yellow-green fluorescence, and it's still being used today. But it wasn't until almost 100 years later, Albert Kuhns, an American physician, really applied these fluorescent dyes toward life science research. He was working on a method to visualize antibodies in a bacterium that was associated with rheumatic fever. And him and his colleagues were able to develop the first method of conjugating fluorescent dye or fluorescein to antibodies to visualize proteins in animal tissue. And we know this technique today as immunofluorescence. And since then, the field of immunofluorescence has exploded. Right? It's pretty much ubiquitous in life science research labs today. If you did a quick search of in PubMed with the term immunofluorescence in the title on an abstract from 1941, you would see over 100,000 publications. And this really is just the tip of the iceberg for fluorescent dyes in research. They're not only just used for labeling antibodies, but specific organelles, membranes, lipids, nucleic acids, and other biological components. And so let's go back to how a fluorescent dye works. So if this is a basic structure of the fluorescein molecule, you can see that it absorbs light at a particular wavelength, and then it becomes excited to an excited state. And then there's some non-irradiative emission, and then ultimately an emission at a longer wavelength than what it was excited by. And so if we looked at a fluorescent dye, within a vacuum, how would we measure the performance? And a key measure is how bright it is. The brighter the molecule, the more signal to noise performance. And this can be measured with a couple of parameters: the molar absorptivity, which is a measure of how much light is absorbed versus at a particular wavelength, and the quantum yield, which is a ratio between how much light is emitted versus absorbed. And the product of these two variables gives you the brightness. But if you're looking at a biological context, right, with a particular experiment with specific parameters, you really can't look at a dye in a vacuum and simply just about how bright it is. There are many, many other factors that you have to consider. For instance, you may need to determine how photostable a fluorescent dye is, depending on how long you're exposing it to a particularly powerful laser. You may have to consider the chemical stability of a dye molecule or how soluble it is. You may have to determine the overall charge of the molecule and how that could affect your signal. If depending on the experimental parameters, you may be worried about the pH sensitivity of a fluorescent dye. And perhaps what many people don't consider is the potential for specific dye structures to form aggregates that ultimately self-quench and diminish your fluorescent signal. So let's go back to your original xanthene-based dyes here. Xanthene here being this core group that sort of defines these particular class of dyes and is the main source of fluorescence for the molecule. And if we go look at fluorescein here, these types of dyes are generally very bright and chemically stable and photostable. But there were some issues with the original fluorescein dye, it being relatively pH sensitive and also really prone to autofluorescence, particularly from tissues, because of the shorter emission wavelength in the green range. And a lot of the autofluorescence coming from tissues also emitted in that particular span. So much of the improvements to the original fluorescein dyes were to actually extend the range to longer red or far red emissions, and that's where we get our rhodamine-based dyes here. And this would entail in sort of increasing or modifying it so that the molecule will be bigger, which generally makes the dye have longer wavelength emissions. And these rhodamine dyes were also a bit more pH insensitive than relative to your fluorescein dyes. So these far red dyes were less prone to autofluorescence and allowed deeper tissue penetration because of the longer wavelengths. However, as I said, when you modify these dyes to make them larger for longer wavelengths, that also makes them less soluble in water, which it does become a problem with these classes of dyes. And there's also still a considerable amount of problematic dye-to-dye quenching during conjugation. So jumping forward to the 1990s, Alan Waggoner proposed a new class of dye, dye that's actually cyanine-based, coming from dyes that were actually used to produce color on film. But he looked at the dye and realized that this base structure could be very useful for life science research applications. So these new class of dyes offer pretty substantial advantages over your xanthene-based dyes. One thing is you'll notice is that they offer a really broader emission window into the longer wavelengths. Xanthene-based dyes being limited to around 600 nanometers, these going from 560 to 800 nanometers. So much more room for longer wavelength emissions, which are generally much better for tissue penetration. And what's also an advantage of these dyes is that these heterocycles that are sort of linked by this polymethylene chain, these are actually more easily able to be conjugated to proteins and other biological components. And you can actually easily modify the emission wavelength of these dyes, to be longer just by altering the length of this polymethylene chain. Increasing the length generally increases the longer wavelength emission. And these dyes were generally much more soluble, offered less dye-to-dye quenching than the xanthene-based dyes. But they do still have issues. They weren't as bright as the xanthene-based dyes. There wasn't as many colors available, and they really didn't have much. They really had poor photostability and chemical stability if you compare them to your xanthene-based dyes. So one solution to tackle the issue of solubility and dye aggregation is sulfonation, which is a modification that's present in today's modern fluorophores, in your side dyes and your hexafluors. And this entails just adding these sulfonated and negatively charged groups around the dye molecules. And this offers a couple of advantages. One, it does improve solubility. The negative charges help with solubility, and it also does help with dye aggregation, and therefore improves biocompatibility at longer wavelengths. But it is a bit of a trade-off. this overall negative charge also leads to increasing nonspecific binding to positively charged moieties, and therefore increasing background fluorescence. And not only that, the added sulfonated groups makes the molecule a bit more hygroscopic, which becomes an issue for conjugation, because these commonly used conjugation reactive groups, NHS and amide, are more prone to hydrolysis. So in designing Biotium's own dyes, Fei Mao and his colleagues really aimed to tackle the issues that were plaguing common fluorophores, including solubility, dye quenching, and also the issue of sulfonation in their new CF dyes. And so let's take a look at the evolution of a cyanine-based dye from the standpoint of sulfonation and the pros and cons of it. So from side 7, as you can see, with some sulfonation, you do get an increase in solubility and less quenching, but you also have an increase in background due to the negative charge. And in the Alexafluor cyanine-based Alexafluor dye, the typical answer was just to sulfonate more, which would lead to, again, an increase in solubility and less quenching, but also more and more non-specific background from the added negative charge. So moving on to a cyanine-based CF dye, what was really novel about this class of dye is a particularly new type of modification for these fluorescent dyes, which is there would be PEGylation or polyethylene glycol, and became sort of the answer to the issues that plagued sulfonation. And so adding these polyethylene glycol compounds, in addition to the sulfonated groups, really offer numerous advantages here. For one thing, they actually help shield the negative charge of the sulfonate groups, and thereby actually helping eliminate a lot of the non-specific background produced by the sulfonate groups. Not only that, they've also actually increased solubility and overall brightness of the molecule. And a bit more subtle feature is that they actually help the photostability of the molecule, and this has a couple of different mechanisms. First of all, the polyethylene glycol sort of makes the molecule a bit more rigid and therefore more photostable, and they also help protect the polymethylene chain from photooxidation from reactive oxide species, so therefore making it a bit more photostable. And you can really see the advantages of PEGylation in the data. In terms of water solubility, if you plotted a graph of absorbance versus wavelength, usually issues of water solubility would be represented by these shoulder peaks that are indicative of non-fluorescent aggregates and due to poor water solubility. And you can see compared to a size 7 and Alexafluor 750 compared to a PEGylated CF750, that this shoulder peak is drastically reduced, indicative of greater water solubility for the molecule. And you can also see it in the overall brightness of the conjugates. If we looked at a relative fluorescence versus degree of labeling in IgG antibodies labeled with fluorescent dyes, you can see that CF750 has a much brighter relative fluorescence compared to Alexafluor or size 7 at the same degree of labeling. And lastly, if you looked at the photostability of the molecule by measuring the absorbance versus wavelength before and after exposure to sunlight, you can see a drastic reduction in the size 7 Alexafluor dyes after 30 minutes of sunlight in absorbance. Whereas CF750, you can see that there's much more absorbance retained after the exposure, and to give proof of the increase in photostability from the changes made by PEGylation. So the chemical advantages of PEGylation of our near-IR CF dyes really translate to a better performance for a variety of applications. For instance, our dyes are generally brighter for fluorescent Western blot or fluorescence. You can see less background relative to a spectrally similar Alexafluor 790 due to the shielding of the negative charge from the sulfonate groups. And this all leads to better in vivo imaging, greater biocompatibility, better solubility, brightness at near IR wavelengths, all really translate to better performance for in vivo work. And our chemists at Biotium didn't stop with cyanine-based dyes. They also looked at the xanthene-based dyes as well. And so let's take a closer look again at our xanthine or our old friend fluorescein and rhodamine. Uh, if we remember, the xanthine-based dyes, they had a very limited emission window of 5 to 600 nanometers. Despite them being very bright, very photostable, and chemically stable, they really couldn't be used past 600 nanometers for far red or and near IR wavelengths, and so that limited their utility. But our chemists were actually able to break this limitation of the xanthine-based dyes, and that actually has to deal with a particularly novel modification to the core structure of the xanthine-based dye. And so if we look at a rhodamine structure here, what the chemists actually discovered that by substituting this benzyl group here with an imidazole group, they can actually extend the emission wavelength by up to 30 nanometers. And depending on the type of imidazole substitution, they can actually extend it up to 60 nanometers. And so this really had tremendous advantages for the xanthine-based dyes. And in addition, the substitution also allowed better solubility and therefore brightness of the molecule. And so this offers several advantages for xanthine-based dyes. Due to their high level of brightness and photostability and chemical stability, and now with the extension of their emission up to 60 nanometers, toward the longer wavelengths, these dyes can now be used for longer wavelengths that are typically reserved for cyanine-based dyes. And so this is really important for applications where a high amount of brightness and photostability are really demanded, particularly super resolution applications like STED and STORM. And again, this substitution also leads to an increased amount of solubility and therefore brightness and performance of the molecule. Just as an example, if you took our rhodamine-based CF680R, which we consider to be the most photostable near IR dye, and is particularly ideal for STORM and STED applications, you can see if you plotted normalized fluorescence versus time and compare the photostability relative to cyanine-based dyes, the CF680R rhodamine-based dye really just is unrivaled as far as photostability in spectrally similar cyanine-based dyes. And it's not just our CF680R dye, but many of our other dyes as well, CF568, 647, 60, really demonstrate superior properties ideal for STORM and STED imaging. Their signal, photostability, and photo switching properties in particular. And many of our dyes, not just listed here, have also been validated for other super resolution applications as well. And for those who are interested in super resolution, particularly for STORM, we have CF dyes labeled that are single labeled secondary antibody conjugates specifically for STORM applications. Our CF dyes have also been validated for another type of super resolution imaging, particularly expansion microscopy, which entails swelling of biological samples in these, or enlarging or expanding these biological samples in these swellable hydrogels, which often leads to diluting the fluorophores. So this particular technique really requires bright photostable dyes, where our CF dyes have also shown excellent performance for. So what does CF and CF dyes stand for? Well, originally, from the earlier patented CF dyes, which were all cyanine-based structures, CF meant cyanine fluorescent dyes. But today that's not really true. Our CF dyes now encompass a wide range of different dye structures. So today we understand CF dyes more as clear fluor, indicative of their superior signal-noise performance. And today we have over 30 dyes. I believe we have around 34 now. We continue to release new dyes that cover a broad range of the emission profile from 350 to 820 nanometers. So now I'm going to talk a bit about sort of the applications of our CF dyes and what they could be used for. And obviously, immunofluorescence is a big application of our CF dyes. We offer a growing collection of CF dye primary antibodies that's in the thousands now. We also offer highly cross-absorbed secondary antibodies. And if for those interested in amplifying the signal with a tyromide signal amplification, we offer a number of standalone CF dye tyromide conjugates, as well as available and ready-to-use kits. And our CF dyes aren't just useful for life science research applications, but also clinical applications. In this example, you can see our CF dyes, a few of our CF dyes were actually used in a 7-color panel for immunophenotyping of colorectal cancer tissue in combination with tyromide signal amplification. CF dyes are also validated for flow cytometry applications as well. In this example, you can see a few of our dyes were actually used in a 40-color panel for cell sorting of human blood samples. Biotium is actually in collaboration with many of the large flow cytometry companies, including Cytec, BD, Thermo, and BioLegend. And we specialize in licensing and developing spectrally unique dyes that fill in the spectral gap of many commonly used fluorophores today. And this is particularly important for developing large multi-parametric panels, particularly for spectral cytometry, where the literal limitation in the amount of colors you can have is actually the amount of dyes that are available. And for those who are looking to label your own antibodies, we offer our mix and stain antibody labeling kits for antibodies, nanobodies, or small ligands. These mix and stain kits are available with 29 different CF dyes as well as other labels, and they can be used for any IgG antibody with a variety of different buffers and additives, including glycerol, BSA, or tris. They're compatible with those components, and they offer really rapid and stable covalent labeling without the need for purification. And this would be particularly useful for multicolor immunofluorescence or flow cytometry applications. And Biotium offers much more than just the CF dye products that are shown here in this presentation. Our gel red and gel green nucleic acid gelsins are safer and more sensitive alternatives to ethylene bromide or CyberSafe. Our NucView caspase scissors are novel substrates for real-time detection of apoptosis. If you're looking at viability, we have 14 different colors of our liver dye fixable dead cell stains that can be used for both microscopy and flow cytometry. And we have many different types of membrane and organelle cell stains. There's just two here, but we offer cell stains for mitochondria, cell surfaces, the nucleus, cytosol, lipid droplets, lysosomes, and other cellular components. And so thank you again, everyone, for listening. I hope you learned something. If anyone is interested in learning more about CF dyes or any of our other technologies, I please encourage you to learn more at www.biotium.com. And thanks for listening.